I don't know.
So, are we all mic'd up? Everybody give a little hello, hello.
Hello?
Hello. No.
Great.
Hello.
So that was, my brain is saturated completely from the morning. I don't know about you guys, but I just feel like I've taken in so much information. And we're going to throw it out to questions in a few minutes. But first, I guess I was trying to find a common thread between all three talks. And for me, it kind of came down to, in a word, sort of performance of the plants and all the different ways that you talked about positive and negative feedbacks, but maybe not in the way that we're used to, in terms of, it doesn't necessarily mean it's bad or good, it's just the way we term it. But we're all looking for ways to make sure that our plants are healthy and that we're growing them in the best possible way to get the results that we want, whether that's to do with the stomata activity or the microbes in the soil or the root systems. I'd love to start with you, Noel, since you were first to talk today. You sort of gave us that amazing whistle-stop tour of all the different underground structures. How do you now use that knowledge in your kind of work when you're designing planting schemes or combinations?
Well, as I was trying to sort of point out in the latter part of my presentation, I think if you understand what is going on underground, you have a much better idea of how a plant will project itself over time, which can vary enormously, how much the structure will spread, but also whether it will stay integrated or whether it will break up. I think that's really, really crucial for when we create a design. We virtually never think about what goes on underground. I'm arguing that if we get what goes on underground right, there's a really good step towards perhaps more stable plantings.
Can you think of an example of where maybe you would have previously used a plant that now that you know more about this, that you go, well, I would use that in a different way?
The Phlomis russelliana in my trial was something, we see that as enormous, great big blocks as you stick it there and it just spreads. But if you stick it there and it's surrounded by plants that are also competitive, or at least stay in a stable block, then it can't do that. So it then has to spread, and then we find that the rhizomes break up, and literally the plant becomes very mobile. It will fragment and move around. And so that was a really nice example. And I think we need to think much more about. I know James and Nigel have always come back to using this word dynamic. We did that book years ago, The Dynamic Landscape, and Bettina Jalgstetter's conference last year was dynamic was in the title there, that there's certain elements within a planting will be dynamic, and we must embrace that element of time when we plan something.
Yeah, and you showed us that sort of mapping project that you did over 7 years. Is there a particular area of research or of mapping like that? What's the next thing you would like to do if you could, or what do you think the thing is that we should be focusing on and doing?
It would be wonderful if we could get a number of collaborators, people with gardens or projects that they know will be there for 5-10 years ahead, and actually do some mapping. Start off with mapping it when it's initially planted, and then return to it. It's relatively simple, but organising it is another matter. But it'd be lovely to be able to do that.
Yeah. And Ellen, you mentioned a couple of villains in your talk, and of course, immediately as a gardener, I wanted to know, what other ones apart from chrysanthemums and potatoes should we maybe kind of be aware of that have that negative plant soil feedback? Is there any others that you discovered?
Well, there's a bunch. It depends on your perspective. It depends really if you want a really species-rich grassland, because one of the key mechanisms and the key purposes ecologically of negative plant soil feedback is to keep that, that species turnover and that kind of high species coexistence going. And this kind of leads a lot of the time to cyclical population dynamics. So, if you have a plant and it drops its seeds and those will be dormant for the first season, it might be in that plant's favour to have a negative plant soil feedback. So then when its seeds finally germinate, it's sort of all ready to go, and it's kind of got that space then available. Talking about time and the life cycle of the plant, there's a bunch of them where it shifts from negative to positive through the life cycle, and one of them is ragwort. And obviously, we probably don't really want ragwort in our gardens and so on, but it is a very important plant, particularly for, pollinators and various things. But that is really nasty, and it's for, in its initial days when it's trying to establish as a seedling and grow, it will have very, very strongly negative plant soil feedback. And so it will kind of create this, because we know it's alkaloids, isn't it, that's full of, pretty nasty stuff. But it will also kind of accumulate a very antagonistic microbial community in the first year or so of its life that will then switch later to quite positive. So then you suddenly see these big mature clumps of ragwort. But yeah, there's loads of species. Cork oak as well is particularly susceptible to Phytophthora cinnamomi, which is quite a famous fungal pathogen. Whereas olives have no response to it at all. So it very much depends on the species and what's kind of there in your area and where you are. So quite context dependent.
Yeah. I know you said there's only been 2 studies done on flooding. There's obviously a lot more work to do there. But again, the questions that sort of sprung to my mind were, you mentioned that the flooding, you thought it might change plant growth form, in the future. What would that look like?
Well, it depends on the duration and the intensity of the flooding. So we know that things like wheat will, within a few hours of flooding, it will create these air tubes in the roots called parenchyma. And you can kind of visualize that in real time over a few hours, that these tubes to sort of increase airflow into the roots. A bunch of plants have it. I think it's a sort of feature of rice as well. So, but they will shift their plant traits and their growth and that kind of thing. And yeah, like they'll close the stomata, they just don't need it anymore, do they? So yeah, flooding has been very much overlooked, which hopefully we're taking steps to rectify. And obviously with no air in the soil, because it's all water, you're probably going to switch to anaerobic microbes, which will change the entire composition potentially of what's functioning in the soil during that time and what's left behind. So that kind of legacy of flooding, quite difficult to know.
And you mentioned salinity being a problem as well. It's not something I think we've come across so much here in the UK in terms of gardens, but how does that work? How does it increase the salinity, and what effects do you think that will have?
Well, you're kind of washing nutrients sort of up from the soil surface. So we have problems with aquifers where if you bleed the aquifer dry in your soils, then that will start to get encroachment of seawater and that kind of thing. There's various risks. More across the world, more in the tropics, I would say, we have big problems with salinity in the soil. But yeah, potentially, and our irrigation systems can be sort of compromised, depending on how they kind of work. But yeah, so.
Yeah. And then Tracy, fantastic talk we just had on stomata. And some of those projects that you mentioned in the end were really interesting. But they did seem to sort of mostly revolve around, very controlled situations where you're doing experiments with light and to do with maybe crops. Can you think of ways, if we were to just wildly extrapolate in the future, into how gardeners and those working in horticulture could maybe benefit from some of those insights you're hoping to get?
I think it's like all research. I mean, I'm talking about that because we're trying to look at crop productivity, but it's a process within all plants. So we're talking about flooding, but if we had grasses or plants that actually didn't shut the stomata, they could be really important for getting rid of water out of flooded areas, because all water, 90%, 99% of all the water on the planet has to go through the terrestrial system, and plants are that terrestrial system. So I think understanding some of them processes can lead to things that, I'm talking about crop research, but also to gardens. I'm a gardener and I sort of think about, I wonder what the stomata are doing there. It also, helps us with water regimes. I mean, we tend to water at night time, so there's plenty of water in the soil in the morning, but that's because photosynthesis is better in the morning. It's the processes that we do them, but this is the understanding behind some of those processes as well.
Yeah. And is most of these changes that you see in the future through gene editing? Is that what
so we've done a lot of, so I showed a graph. We also look at natural populations. There's lots of natural variation out there, and especially with stomata, because they're changing we're linking that stomata with plant performance, with photosynthesis, and that changes depending on the plant, the environment, the soil will change all of that, change the number of stomata, the size of the leaves, all of that will change how much water has been lost through those plants and how much water is needed for them. So, yeah.
I was fascinated to hear about the Carbon Garden project with Beth Chatto. I'm just wondering how much time we have. I'd love to hear a little tiny bit more about that.
Yeah, so we've been looking at lots of plants around the garden and plants that the nurseries have as well, and we've been trying to measure how much carbon they're taking up. And this came from the fact that, we're all very sort of conscious of our carbon footprint. And I was chopping a tree down in my garden one day, and I thought, God, look how much carbon I'm sort of just chopping off this tree. And then I thought, I have a lot of house plants, I have a lot of garden, how much am I sort of, sequestering some of that carbon? I know there's a cycle for that carbon, but it made us think about what sort of types of carbon gains by different plants. So we're now sort of measuring the photosynthesis and the stomatal behavior on some of those plants and looking at how much carbon they're taking up to see what different ones are doing. And usually that's translated into growth, but not always growth that you see. It could be growth underground, into the rhizomes, into the rhizosphere. You said 40% of the photosynthate. I used to think 20%, so there's a lot of photosynthate going there. So we're trying to just see what them different carbon footprints are like in different... would you buy a plant if it was more sequestering more carbon? And it's a perennial.
That's amazing. I'd love to throw it open to questions from the floor first.
Yeah, quickly. I think there's a big question about plant society here, and just like your views on that, because each of us, gardeners want a particular plant society in terms of a selection of plants. Now, in ecology, there's been, a long debate which goes back, like a long time, which is called phytosociology, which was, that society of plants. And so there's schools which, debated whether plants were individuals or part of an association. So people like Blom-Bronquet and people like that in the past. That's epitomized today in, the National Vegetation Classification. So I'd just like your views in terms of, like, soil obviously is critical. You could apply the same reasoning to what Merlin was saying in terms of associations or individualism. Is it the selfish gene? Are we looking for the ways that we can look at these things in a different way? I'd just like your opinions on that.
Well, I think the future will very much be about novel ecosystems, partly because of alien species that establish, aggressively or otherwise, and climate change. So everything really is up for grabs, and the ecologists have traditionally been very fixated on so-called natural plant communities, which in many cases are anthropogenic anyway. So I think we need a lot more work on novel ecosystems, how they can be biodiverse, how they can be stabilized, how we can use them aesthetically. It's interesting to hear about a few places or people are beginning to be less worried about invasive species and beginning to think about the species they're stuck with as part of novel ecosystems, how they can be incorporated into designed landscapes.
Okay. Humira?
Hello. I was just trying to understand the plant soil feedback, because you were saying that potatoes, say, take 7 years and they have all these particular things that come with them, but with drought, it takes it back to 0. So could we kind of recreate droughts to then plant potatoes every 2 or 3 years? What would be
Interesting.
Humira might have come up with a global fix.
Hunger. Brilliant.
Well, I suspect what you'd get is microbial dormancy, that as soon as you alleviate the drought, it would just be back, like it's not going anywhere. So, yeah, I mean, the drought study that I did was a couple of species. So, yeah, by no means, general principles. But, yeah, I mean, I think things like Phytophthora, they have a long legacy, particularly because they have, this, the mycelium and the fungal hyphae, they're quite hardy to drought. They just come back. So, yeah, tricky.
we're facing a very hard challenge in our cities, and especially like I work and live in Barcelona, and talking about stomata or listening about this interesting research, we have for one side we have to use every time more drought tolerant species. But in the end, also for the cities, we need the cooling effect, so I don't know if there are specific research or something that is going to work with this kind of contradiction in terms of what we need from plants for urban environments.
Yeah, I mean, I think that's sort of some of the stuff that we still don't understand how some of these processes work, and I think it's by understanding them that you can then maybe develop plants for the, for some of those environments. So, for example, like our speedy stomata, if they're fast, it means that then you still need that evaporative cooling, but if you can do that evaporative cooling when you actually need it, and they're more in tune with the environment, that's of course highly dynamic, it means that you can open the stomata when you can get the CO2 or when you need to cool, but then if you shut them fast, you'll save that water in the plant itself, but also not be repleting the ground reserves to try and make it more available for when it might be more beneficial. So say reproductive phases or something like that in a plant. Does that sort of sense?
So we got a couple of questions from people watching on the live stream. Noel, do you have a list of recommended plants for a volunteer group planting in public spaces that are low maintenance and self-repairing? Easy.
Well, I'm not going to read out a plant list, but simply suggest that the next issue of the Hardy Plant Society Journal, and I expect many of you are hardy planters, contains an article which is a sort of a summary of my presentation today. And that ends up with a nice worked out example of a planting that I think will work.
Yes, you did just flash up those plant names. I didn't even have time to get my phone up. So there you go, Hardy Plant Society Journal. The next question is for the whole panel. We did touch on it just a second ago, but what impact might salination of soils following flooding have on planting schemes in sustainable urban drainage and flood retention landscapes?
We already have this problem with salt from de-icing. And certainly I know Svante Duttweiler in Weinsteffen in Germany has done some work on that, some very useful lists of species. You could probably dig that out on Google Scholar.
2 minutes left. Everyone okay? So we have time for another question. Yeah, Eddie?
Thank you. Just on the potato issue. So there's some really brilliant work by a French agronomist called Oliver Hussin on the redox potential. And we concentrate as gardeners a lot on pH, but we don't talk about electrical potential and redox potential. And it really is worth looking up his work because there is a distinct relationship between pH and EH. And when you map pathogenicity, you find that there is a pattern. And whichever plants are infected, they are infected because of this pattern of this relationship between redox potential and not. In a nutshell, redox potential is like the energy of the electrons. Electrons run all life. So if you plough a soil, get lots of oxygen in, then you've got high redox potential. If it's compacted, then it's very low redox potential and the electrons can't do anything. And there's this middle ground where the pathogens work. So as gardeners, if we can aim to get our soil conditions and the sap of our plants in this zone, which is outside of the pattern of pathogenicity and insect herbivory, then you will not get any of those diseases. Really well worth looking at his presentations. Oliver Hussin, redox potential.
Is it cost effective? Would it be cost effective to maintain your pH within this?
Yeah,
bound?
I've been using it in my garden I was pretty much the only person who avoided Phytophthora last August when it was wet and humid at the same time, and I just looked at the weather patterns and I thought, yeah, Phytophthora, yeah, okay, well, tell you what, I can't change the temperature, but I can shield it from the rain and keep that potato patch in August a little bit drier. So effectively I was changing the EH, the electrical potential, and only one plant on the edge of my patch got Phytophthora, the rest didn't. So I kept those plants out of that pattern. Yeah. worth looking at.
I think we'll have probably more questions for you on that idea when you do your panel later today. That's wonderful. Thank you so much, everyone. And it's lunchtime. Yay!